Welcome dear students. I am Dr. Suraj, Assistant Professor of Botany and HOD of Department of Botany of Barakpur Rashtraguru Surendranath College. In my previous class, I have discussed about photosynthesis. Today, I will discuss about respiration, mainly plant respiration. During photosynthesis, we saw that photosynthesis provides the organic building blocks that plants and nearly all living organisms depends on. And we know that during photosynthesis, solar energy is transformed into chemical energy, and this chemical energy in the form of ATP is transient into this organic building block, or you may call carbon compound. In respiration, what happened? In respiration, release of energy stored in carbon compounds in a controlled manner for cellular use will be happened. So, in respiration, the energy which stored in carbon compound will be released here, and of course in a controlled manner, and this energy will continue other cellular reaction. Now, come to plant respiration. Actually, we know that respiration is of two types. One is aerobic and another one is anaerobic, that is presence of oxygen or not. I want to say here that the outline, the basic outline of respiration in plant or in animal or in any other eukaryotic organism is more or less same. Some basic difference in enzymatic process is there, but the basic outline is same. What happened in aerobic respiration? Aerobic respiration is the biological process by which reduced organic compounds are oxidized. Okay, reduced organic compounds will be oxidized here in a controlled manner. Okay, and here energy is released and transiently stored in a compound, that is adenosine triphosphate, which is used by the cellular reaction for maintenance and development. So, respiration is a very important biological process because the energy will be released in this process. Now, come to here. Actually, glucose is substrate for aerobic respiration in most plant cell types, but other carbon, that is disaccharide sucrose other sugars, organic acid, trisphosphates from photosynthesis, and metabolites from lipid and protein degradation are also maybe the substrate, or they act as a source of substrate. Look at this diagram. It is the overall reaction of respiration. Actually, mainly four biological process will be happened in respiration. First one is glycolysis. Second one is pentose oxidative pentose phosphate pathway, and third one is citric acid cycle, and fourth one is oxidative phosphorylation. So, these are the four basic reaction steps through which plant aerobic respiration will be happened, and three cellular organisms one is plastid, second one is cytosol or cytoplasm, and third one is mitochondria. So, these are the three organelles: plastid, cytosol, and mitochondria. These are the three cellular organelles where respiration will be taken place. Now, come to the first point, that is glycolysis. Actually, what happened in glycolysis? In glycolysis, sugars through a series of reactions will be converted into hexose phosphate, then 2-trisphosphate, and finally it will be converted into organic acids. The organic acid is pyruvic acid, and this will be happen in cytosol. Another one, pentose phosphate pathway. It will involve in plastid and cytosol. And what happened here? Here 6 carbon compound will be converted into 5 carbon compound, and carbon dioxide and NADPH will be produced. And citric acid cycle. Actually, most of the energy from sugar, oxidation of sugar, will be released through this cycle, through this citric acid cycle. And last one is oxidative phosphorylation. Okay. Now come in details. Glycolysis, actually glycolysis involves, I have already mentioned, a series of reaction catalyzed by enzyme located both in cytosol and plastids. For example, a sugar, maybe a sucrose, that will be partially oxidized here, not fully oxidized, will be partially oxidized here. And what happened here? 6 carbon sugar phosphate, I have already mentioned a hexose phosphate, that will be transformed into 2,3 carbon sugar phosphate, that is triose phosphate, and finally it will be converted into an organic acid. This organic acid is pyruvic acid. And here release of small amount of energy as ATP and reducing power NADH will be took place here. Okay. So small amount of energy will be released here. In the second step, that is in oxidative pentose phosphate pathway, what I have already mentioned, that in oxidative pentose phosphate pathway, which is also located in cytosol and plastids as glycolysis, what happened in oxidative pentose phosphate pathway? In oxidative pentose phosphate pathway, a 6 carbon glucose, 6 carbon, that is glucose 6 phosphate, will be initially oxidized to a 5 carbon, and that is ribulose 5 phosphate. So in oxidative pentose phosphate pathway, it will happen that a 6 carbon glucose 6 phosphate will be initially oxidized to 5 carbon ribulose 6 phosphate, and 1 carbon will be lost as in the form of Ca2. As I have already mentioned here, look at here, release of CO2 will be happened here, and reducing power will be formed in the form of NADPH here. In the third process, that is in citric acid cycle, which is also known as TCA cycle. So what happened in citric acid cycle? During glycolysis, the ultimate end product is an organic acid, and that organic acid is pyruvate. And now this pyruvate will be completely oxidized to CO2 in citric acid cycle. So in citric acid cycle, the end product of glycolysis, pyruvate, will be completely oxidized to CO2, and that will be controlled by various enzymes. And citric acid cycle is happened in the innermost compartment of the mitochondrion, innermost compartments of the mitochondria, that is the matrix. Okay. So, matrix, mitochondrial matrix is the location for citric acid cycle. And of course, this process will mobilize a major amount of reducing power. As I have already mentioned earlier, okay, here 16 NADH, 4 FADH2 will be produced, and a small amount of energy in the form of ATP or GTP will be produced from the complete breakdown of sucrose molecule. Okay. And in the fourth step, that is in oxidative phosphorylation. Okay. In the fourth step, that is in oxidative phosphorylation, what happened here? Here actually electrons are transferred along an electron transport chain consisting of a series of protein complexes which is embedded in the inner of the two mitochondrial membranes. So, citric acid cycle and oxidative phosphorylation both will happen in mitochondria, one in mitochondrial matrix and this oxidative phosphorylation in the inner two mitochondrial membranes. Now, actually what happened during oxidative phosphorylation? In oxidative phosphorylation, actually this system transfers electrons from NADH to oxygen. Okay. Electrons will be transformed here from NADH to oxygen, and this will transfer release of large amount of free energy. So, in short, these are the four reactions of respirations what I have mentioned here. Now, in this lecture actually I will discuss about two reactions. One is glycolysis process and second one is oxidative pentose phosphate pathway. You can see here I have already mentioned that where in the cell glycolysis happened, it is plastid and in cytosol or cytoplasm. The Greek word glycolysis, the Greek word glykos means sugar and lysis means splitting. So, actually in glycolysis sugar, it may be sucrose or in any other form that will be transformed or converted in the end product, which is pyruvate, and also a little energy, a little amount of energy in the form of ATP and NADH will be released here. So, come to here the details. What happened? In the early steps of glycolysis, carbohydrates are converted into hexose phosphate, each of which is then split into 2 triphosphate. So, first carbohydrate will be converted into hexose phosphate, then this hexose phosphate will be split into 2 triphosphates, and in a subsequent energy conserving phase each triphosphate will be oxidized into an organic acid, which one is pyruvic acid. Okay, pyruvic acids. Come to this portion here, this portion. When molecular oxygen is unavailable, then what happened? Such example, that is in plant roots in flooded soils. Here glycolysis can be the main source of energy for cells. Okay, glycolysis, pentose phosphate pathway, citric acid and oxidative phosphorylation. These are the 4 way, or these are the 4 biological process of respiration. But when molecular oxygen is unavailable, there glycolysis can be the main source of energy for cells. Remember that. Now come to the reactions of glycolysis. This is the first phase of glycolysis. And this initial phase of glycolysis, what happened in this initial phase? Look at here. Through photosynthesis, starch will be stored in the chloroplast. This starch, through a series of reaction, will be converted into glucose. Okay, this starch will be converted into glucose and transferred into cytosol. And here glucose will be converted into glucose 6-phosphate, then fructose 6-phosphate, and then fructose 1,6-bisphosphate, and finally glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So 6 carbon glucose 6-phosphate will be converted into 2 triphosphate, this glyceraldehyde 3-phosphate. This is the one way of initial phase. What will be the in the another way? Sucrose, which is present in the cytosol already. Sucrose, which is present in the cytosol already, will be converted into fructose, or it will be converted into fructose and glucose. Sucrose will be converted into fructose, glucose, or UDP glucose. Then fructose will be converted into fructose 6-phosphate. Okay. Then this fructose 6-phosphate will be converted into fructose 1,6-bisphosphate and then glyceraldehyde 3-phosphate. This is a triphosphate. So here also 6 carbon. Now these are the reaction steps which is known as energy conserving phase of glycolysis. So the first one steps, which is known as initial phase of glycolysis, where substrate from different sources will ultimately converted into triphosphate, and now this triphosphate will be converted into pyruvate. Okay, this triphosphate will be converted into pyruvate, and these steps of glycolysis are energy conserving phases. Now, what happened here? You can see here that the glyceraldehyde will be converted into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate will be converted into 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenol pyruvate, and finally pyruvic acid or pyruvate. Look at here, from these steps to these steps, that is glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, 1 NADH, that is reducing power will be produced. Look at also here, from 1,3-bisphosphoglycerate to 3-phosphoglycerate, and look at these steps, phosphoenol pyruvate to pyruvate. These two steps are also producing energy in the form of ATP. And interestingly, these steps are known as, and this type of phosphorylation is known as substrate level phosphorylation. Why substrate level phosphorylation? Because one phosphate will be given or will be taken from the substrate, that is from this substrate 1,3-bisphosphoglycerate, that is substrate of a reaction is the source of phosphate. That is why this is known as substrate level phosphorylation, this step and this step. Now, pyruvate, what will be happen at pyruvate? Pyruvate ultimately may produce pyruvate in the mitochondria will be happened here what? In mitochondria it will go in citric acid cycle. In another way it will ultimately converted into ethanol through fermentation reactions. Look at here, this phosphoenol pyruvate may be converted into oxaloacetate, and this oxaloacetate to malate, and ultimately malate will be transferred into mitochondria and it will start citric acid cycle. So, these are the steps of glycolysis. Now, what are the enzymes involved in glycolysis? For the initial phase, look at here, from sucrose, inverter enzyme will convert sucrose into glucose, and hexokinase will convert into glucose 6-phosphate. Glucose 6-phosphate to fructose 6-phosphate, the enzyme is hexose phosphate isomerase. Okay. And ATP dependent phosphofructokinase will convert these steps, will convert fructose 6-phosphate to fructose 1,6-bisphosphate, and enzyme aldolase will convert fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. Okay. So, initial phase are controlled by these enzymes. Look at here, these steps here one ATP molecule will be required, and also these steps will require one ATP molecule. So, in initial phase 2 ATP molecules will be required to carry out reactions. Now, come to the energy conserving phase. In energy conserving phase, look at here, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, this reaction will be carried out by glyceraldehyde 3-phosphate dehydrogenase enzyme, and 1,3-bisphosphoglycerate to 3-phosphoglycerate, this reaction will be carried out by phosphoglycerate kinase, and 3-phosphate to 2-phosphoglycerate, phosphoglycerate mutase, and from 2-phosphoglycerate to phosphoenol pyruvate, it is enolase, enzyme is enolase, and from phosphoenol pyruvate to pyruvate, the enzyme is pyruvate kinase. So, these are the all enzymes which are involved in glycolysis process. Next, I have already mentioned about substrate level phosphorylation. The substrate level phosphorylation, which is involves the direct transfer of phosphate group from a substrate molecule to ADP to form ATP. So, this is the substrate level phosphorylation. Actually, plants have alternative glycolytic reaction, that is gluconeogenesis. And what happened during gluconeogenesis? The glycolytic degradation of sugars to pyruvate occur in most organisms, but in many cases it is observed that what happened, but many organisms can also operate a similar pathway in the opposite directions, that means from organic acid to sugars. Okay. This reaction is known as gluconeogenesis. So, mainly glycolysis, in case of glycolysis, sugar to organic acids, but in many other cases organic acid to sugar also happened, and this reaction is known as gluconeogenesis. Now, come to the second part of respiration. This is the oxidative pentose phosphate pathway. I have already mentioned that during oxidative pentose phosphate pathway, in the first two steps of this reaction, 6 carbon, that is hexose phosphate, will be converted into 5 carbon ribulose bisphosphate, and this oxidative pentose phosphate pathway is also known as hexose monophosphate shunt. Okay. Come to the reaction point. This is the reaction point. What happened here? Look at here. During the first two reactions, what happened? Glucose 6-phosphate first will be converted into 6-phosphogluconate. Okay, 6-phosphogluconate. So, glucose 6-phosphate will be converted into 6-phosphogluconate, and then from 6-phosphogluconate to ribulose 5-phosphate. This is the first two steps, and during these first two steps, what happened? One carbon dioxide will be released, and just look at here, here and here, both the steps will produce reducing power in the form of NADPH. Next, in the next steps, what happened? In the next steps, the ribulose 5-phosphate will be converted to glycolytic intermediates, that is into fructose 6-phosphate and glyceraldehyde 3-phosphate through a series of metabolic interconversion. Now, what are the conversions? First, in one way, ribulose 5-phosphate will be converted into ribose 5-phosphate, then glyceraldehyde 3-phosphate, and ultimately fructose 6-phosphate. This is the glycolytic intermediate. In other reactions, ribulose 5-phosphate will be converted into xylulose 5-phosphate, then cedoheptulose 7-phosphate, and then erythrose 4-phosphate, and finally glyceraldehyde 3-phosphate. So, this is the reactions of oxidative pentose phosphate pathways, and importantly NADH and more other precursors of other synthesized organism or will be produced here. Look at this one. The net result is the complete oxidation of one glucose 6-phosphate molecule to CO2 is the synthesis of 12 NADPH molecules. 12 NADPH molecules. And what is the importance of that? Importance of oxidative pentose phosphate pathway is it produces NADPH and biosynthetic intermediates. Okay. So, oxidative pentose phosphate pathway produces NADPH, which is reducing power, and other biosynthetic intermediates. Now, lastly come to here actually how oxidative pentose phosphate pathway will be regulated. It can be regulated through redox regulated. Okay. The oxidative pentose phosphate pathway is redox regulated. Look at here. Each enzymatic step in oxidative pentose phosphate pathway is catalyzed by a group of isoenzymes that vary in their abundance and regularity properties among plant organs. The initial reaction of the pathway catalyzed by glucose 6-phosphate dehydrogenase is in many cases inhibited by high ratio of NADPH to NADPH. So, high ratio of NADPH to NADP plus can regulate oxidative pentose phosphate pathway. Again, in the light, little operation of the pentose phosphate pathway occurs in chloroplast. So, light is another. Glucose 6-phosphate dehydrogenase is inhibited. Look at here, glucose 6-phosphate dehydrogenase is inhibited by a reductive inactivation inactivation involving the ferredoxin thioredoxin system and by the NADPH to NAD plus ratio. So, this is the regulatory way or how oxidative pentose phosphate pathway will be can be regulated. Okay. So, this is the all of glycolysis and pentose phosphate pathway. In my next class, I will discuss about citric acid cycle and oxidative phosphorylation. Thank you.